Thank you very much, Christoph, for your introduction. So since my colleagues already introduced you to the amazing BCI world, I will start with a brief introduction on functional near-infrared spectroscopy. Basically, fNIRS is a non-invasive optical imaging technique that indirectly assesses neural activity by measuring changes in oxygenated and deoxygenated hemoglobin in the brain tissue using lights of two different wavelengths, which are 760 and 850 nanometers. There are 6 different ways for infrared light to react with the brain tissue. One of them, the first one, is the direct transmission, the second, diffuse transmission, specular reflection, diffuse reflection, scattering, and absorption. fNIRS focuses primarily on absorption. Differences in the absorption spectra of deoxyhemoglobin and oxyhemoglobin allow the measurement of relative changes in hemoglobin concentration through the use of light attenuation at multiple wavelengths. 2 or more wavelengths are selected with one wavelength above and one below the isobestic point of 810 nanometers, at which deoxyhemoglobin and oxyhemoglobin have identical absorption coefficients. Using the modified Beer-Lambert law, relative changes in concentration can be calculated as a function of total photon path lengths. So typically, the light emitter and detector are placed ipsilaterally, meaning that each emitter detector pair are on the same side on the subject skull. So recorded measurements are due to backscattered or reflected light following elliptical pathways. fNIRS is most sensitive to hemodynamic changes, which occur nearest to the scalp, and these superficial artifacts are often addressed using additional light detectors located closer to the light source, the so-called short separation detectors that I will present you a bit later. At this moment, there are 3 modalities for functional near-infrared spectroscopy. The first one is continuous wave. The second one is frequency domain and the time domain. In today's demo, we will use the continuous wave technique that uses light sources with constant frequency and amplitude. Many continuous wave fNIRS commercial systems use estimations of photon path length derived from the computerized Monte Carlo simulation and physical models to approximate absolute quantification of hemoglobin concentrations. Due to their simplicity and cost effectiveness, continuous wave fNIRS is by far the most common form of functional fNIRS, since it's the cheapest to make, applicable to with more channels, and ensures a high temporal resolution. Coming back to the raw signals, today we will use the oxygenated hemoglobin, HBO, that we can see in the first plot with red, the deoxygenated hemoglobin, or HBr, in the lower plot with blue. And usually the signal-to-noise ratio is generally better for HBO. But in both plots, there are clear visible pulsatile fluctuations, which represent a quality indication. Okay. So in this figure, you can see the average oxygenated hemoglobin responses for a motor imagery experiment. The dotted line in here represents the task onset, so the moment when the user is instructed to start imagining a limb movement. The task duration, it's about 10 seconds. The initial dip, it's more or less 2 to 3 seconds. The peak, it's at about 8 seconds. And then we can see the undershoot. In the same way, we can see the deoxygenated hemoglobin response for the same experiment. So again, the task onset, the negative peak in here at about 8 seconds. And yeah, again, the same task duration of 10 seconds. As a question, why combining fNIRS with EEG? Because it uses an additional source of information. It improves the performance, the BCI performance, and also the robustness, neurovascular coupling, and fNIRS, it's a very good complement to an EEG system. All the experiments with EEG, fNIRS, and so on require a succession of tasks to be mentally performed by the user. For proper task execution in terms of timings, our team developed the paradigm presenter software that allows researchers to create their own paradigm. So Francisco, my colleague, already showed you some paradigms while my colleagues were doing something directly in HISIS. So paradigm presenter allows you to define and to present high precision stimulus, define logical task sequence, adding different stimuli to tasks, present task and stimuli sequence, and provide real-time software trigger. So let's say we have a logical task sequence. This is the paradigm. We define a single task, the second single task, or multitasks that do contain some other single tasks. Okay, so we can see the numbers. So this is the task 1, task 2, 3, and 4, and so on. Basically, all these tasks are coded, all these trees coded into a XML file. Okay, so these are the stimuli tags, very simple one. So first, we start adding the duration in seconds for every single one. So let's say the first stimuli, it's the caption hello. The second one, it's a picture, which is, I don't know, we are setting it to last for 0.5 seconds on the screen. The third task, it's again another picture. And the last task, it's a caption with bye. Of course, also movies can be presented, so you can insert a movie in here and it will be properly displayed. So you can record both EEG and also fNIRS while watching that movie. Going to the NIRS device, today we will use the NIRX device, the NIRS port 2. The fNIRS contains multiple channels, so today I will present you 8 times 8, so 16 channels, 8 sources, and 8 detectors. But my current setup can go up to 32, so 16 times 16. EEG is more the add-on, and of course there is the NIRX has a powerful fNIRS analysis software available. The NIRS Port 2 device, it's a high-powered dual-tip LEDs. It can go up to 80 sources and 80 detectors while having 5 devices synchronized, of course. It allows wireless or USB connection and up to 240 Hz sampling rate. Earlier, I discussed something about the short channel detectors. So the short channel detectors in NIRX, pick up the extracerebral signals close to the source and route these signals to an available detector channel through miniaturized fiber optics. In here, in the upper picture, we can see the respiration, cardiac, Meyer waves, and the functional brain activity. In the lower left plot in here, we can see the contaminant signal. The lower right plot shows the clear HBO and HBR signals from which the above shown non-cerebral signals were subtracted. In terms of software integration, I think my colleague Frankie also mentioned. Right now we provide recording EEG and fNIRS with G recorder and with G HISIS for our amplifiers. Recording with Aurora software for NIRS Port 2. And the synchronization is done via lab streaming layer or TTL triggers. In here, as a first example, we have the paradigm presenter window and the Aurora while recording fNIRS. So we can see that we have the a right arrow, which means in the motor imagery experiment, the cue for start imagining the right hand movement. And we can see the marker in here in Aurora, sent via LSL. In here we can see the left hand cue and also the marker in Aurora. And here I switched the plot to multiple channels to see every single channel in a separate plot. So we can see that the marker in here it's blue. Again, with G recorder that records the EEG. My apologies. Here is the EEG signal, and we can nicely see the trigger, exactly the same as in Aurora. And just a small conclusion here that G recorder and Aurora software run independently. The paradigm presenter runs the paradigm and stimuli, sends the trigger via UDP to G recorder, sends the triggers via LSL to Aurora, and very important is that these triggers are sent simultaneously. Some other advantages, the EEG data is available in G recorder native format as HDF5, while fNIRS data is available as sNIRF. fNIRS and paradigm presenter can run, fNIRS and also G recorder can run on the same machine simultaneously or on different machines. And this is very, very important. Regarding G.HISYS, we have exactly the same implementation. In here, the triggers are sent into the EEG in this plot via LSL. And also the on the fNIRS scope. HISYS records at the same time both EEG and fNIRS. So Aurora runs in parallel with MATLAB Simulink and the G.HISYS model. The paradigm presenter is integrated in HISYS. The HISYS sends triggers via LSL to Aurora. Aurora distributes fNIRS data via LSL. G.HISYS synchronizes EEG, fNIRS, and the paradigm. Real-time control of fNIRS in G.HISYS is possible. And we do have the opportunity to work with fNIRS data under MATLAB or under its native sNIR file from Aurora. In here we have a motor imagery experiment, the paradigm. So usually we have couple of seconds for pre-resting, in this case 30 seconds. We have the instruction that keeps for 2 seconds, the task that has to be done by the user, which will last 10 seconds, and then the user has to rest. So for a number of trials, in this case, 50 trials. The experiment setup in here, so we have the G.Nautilus and the fNIRS device. We have the computer and 2 functional electrical stimulators that can provide stimuli to the hand. So while the user imagines the left hand movement, this neurostimulator induces the hand opening movement for the left or for the right hand, depends on the imagined movement. So only if the user imagines correctly the left hand movement as instructed by the cue, only that's the moment when the neurostimulator will start stimulating the hand. This is the HISYS model, the Simulink model that we will use today for recording both EEG and fNIRS. But up to here, talking is talking, doing is doing. So I will start now setting up the cap on my colleague, Alex. So I took the freedom first to set up the cap because, yeah, there are many electrodes. So there are 16 optodes, 8 sources and 8 detectors, and 16 EEG electrodes. So I took the freedom to pre-mount them all because it will take a bit of time. But I will just remove 1 or 2 of them to show you different ways and how easy it's to set up everything. But first, Alexandru, please, let's switch places. Please have a seat. So I will start now in mounting the cap. First thing, as also my colleague Francisco showed you earlier, it's nicely mounting the cap on the user's skull. It's very important that the CZ electrode is exactly in the middle of the distance between the nasion and the inion. So after years of experience, I can measure it like this. And also, it has to be on the center line in between the preauricular points. Okay, so this would be the first step in setting up the cap, which is hybrid fNIRS with EEG. The optodes. I will take the camera and I will bring it a bit closer. So these are the sources with red and detectors with blue. When mounting them, so this is a short source. We just have to put in here the electrode, the source, and then put a holder on it. Of course, this is done via a montage in Aurora. So Aurora software for recording fNIRS will show all this. But when placing all these sources, I will remove it again. It is very important that this dual-tipped source has to properly touch the skull. As we see in here, he has some hair. So this hair has to be...
Dano, can you actually hear me?
Yes.
Would you mind to shut down your presentation, because then we could see the video full screen?
Yeah, sure.
Then you can connect again. Yeah, full size. Thank you.
Full size, okay. So coming back, this is the source. Okay, let me see how I can properly. Okay, that's better. So this is the source of toad. So in here we can see that there's a bit of hair, and I'm just removing it, putting it to the side, and place the source in here, and then the holder that has a small spring inside. And I will show you in a bit what's about all this, how to optimize all the signals. There are different holders, because if the signal is pretty bad, you can first of all make something like this with the electrode, with the holder, so the tip of the optode gets to the skin. If it's not working like this, you can switch this holder with another one. So there are all these holders have a number in here written. So this is number 2. And this is with respect to the spring strength. So number 2 is stronger. There are 4 different types of holders. So from 0 to 3, which 3 is the most powerful, the strongest spring. Okay, I will put this back. And it's very easy, just flip it. Okay. So first I will start also the Aurora. I will start sharing the screen again to check the signals. So this is the Aurora software. First of all, in here, we do have to select the montage. So I will select it. And this is the montage that Alex has on his head. So we have 4 sources on the screen sharing has stopped. Okay. Let me start sharing it again. Sure. Okay. And let me move this bar from here because it. Okay. So this is the montage that Alex has on his head. With red, we have the sources. With blue, we have the detectors. So first thing that we do have to do is to check the signal optimization. Yes, continue. And now the device is checking the signals in between the sources and the electrodes. So you can see in here some paths. So if they are all green, everything is okay. Yellow is acceptable. Red is critical. So we can see that we do have a couple of them critical. So in between source 1 and D1. And as I told you earlier, I will start playing a bit with them. So they do touch the skin. Okay. And the other one was in here, between these two. He has a lot of hair, I must admit. Okay, continue. Let's see again if the signal is better now. If not, as I said, I will have to remove the holder and do it again, and remove the hair from there. Okay, now all these signals are acceptable. Okay, good. Next, I will start adding the gel to the EEG electrodes. Now let's assume that with fNIRS, all the channels, everything is okay. So I will start gelling all the electrodes. So I added, as I said, 16 electrodes on the motor cortex, on his head. Yeah, this procedure takes a couple of minutes, 2 or 3 usually. I think Frankie also mentioned earlier that you have all the electrodes. Usually you have to start filling the electrodes with gel from left to right, because they are also numbered like this, 1, 2, 3, 4, starting from left to right, from the first row to the last one. Okay. Usually for coming back to fNIRS, to setting up the electrodes, the curly hair is the biggest problem. But as I said, using a spring number 3 and removing the hair first, it will work properly. Also, my colleague Alex has a lot of hair because I already emptied my syringe gel. So he'll be very happy after this presentation. Okay. There's a bit of gel also on his shirt, but probably he won't be mad at me after this. Okay. Ground electrode. And the reference for the right earlobe. Okay. Now I will start G recorder to check also the EEG signal. G recorder, open. Let's wait a bit until it opens. Yes, but I forgot to start up the amplifier. Okay, it's done. Okay, and I will select the device. EEG Nautilus. This one here. Okay. Let's go to settings. So I will apply a bandpass filter on all channels in here. So it will be a wide bandpass filter in between, let's say 0.1 to 60 hertz to see a bigger, a higher band. Okay, apply to highlighted. And we'll apply also a notch filter, 40 to 52 hertz. Apply to highlighted. We can see them in here. It's okay. But I forgot to acquire all signals. Okay. Okay, channel 16. All channels and have a first look on the EEG signal. And I will auto scale all these channels. Okay, on the third channel, probably I got to add some gel. So which is 1, 2, 3, this one here. Okay, now it's a bit better. It's a very easy way to investigate signals only while looking them. Of course, we can check the impedance and so on, but that's a different story. So from my point of view, the signal looks very nice. Okay, so I will stop it for the moment. Yeah, please start crouching your teeth a bit. Strong, okay. Start blinking with your eyes. Nice. Please close your eyes and just stay still. Yeah, we can see the alpha, especially in the front channels. Yeah, thank you very much. So I will stop it for the moment and go to Paradigm Presenter and to Aurora first. So we already did this parts in here. We've checked the fNIRS signals. Let's go to back to the montage to set the LSL parts. So we have the Paradigm Presenter here. Oh, sorry. And I will load the paradigm. Let's say it's on desktop. fNIRS paradigm, motor imagery. Okay, so I will start it for the moment so we can receive everything on Aurora. Okay, so I will edit it. We'll go to LSL stream names, and I have to check in here the data of stream name that it's coded directly in the paradigm XML, the triggering stream name, and the feedback trigger number, the fallback trigger number, which is 1 and 2. Okay. And I will test the connection here to see, okay, we got this message saying that we do have the trigger on LSL. We'll click on OK, save it, and we'll load it again. Okay. And I will go directly to recording. So I will start the record. Yes, I want to start the measurement because we already checked the signal. And now, so this is the whole channel view in here in Aurora. In a bit, we will see the triggers coming from the paradigm presenter as soon as we'll have a arrow pointing to the right or to the left. So to the right, this is the marker saying that, okay, the user imagined right. The user has to start imagining the right hand. At the same time, I will go back to the recorder. I will make it a bit smaller and also Aurora. And I will start the measurement again, just to see the triggers. So as soon as I clicked on it, also the paradigm presenter should a trigger in here in the G recorder. Okay. So as I said, G recorder saves all this EEG data in HDF5 file format, and Aurora at this point saves everything into fNIRS format. G recorder doesn't record fNIRS, but as I said, we have it in its native form. Going back to Aurora, I only tested the triggers now, but we have different displays. So we have the line. Of course, in here we can switch the scale, higher, bigger, and so on. And you can see in this type of display, it's pretty difficult to see something. So we have the line plot. In here, every single channel, it's individually. So we have the HBO and HBR for every single channel. So what you see in here, these oscillations in HBO, basically it's the ECG, it's the heart rate. So it's very nicely seen. This means that the signal is good. As I told you earlier, it's a very good signal indicator. Okay. Basically, we have it topographically. So if I go also in this way here, I can see, if I point the mouse on this line, I can see the signal for this line. Okay, HBO and HBR, how they are moving. Okay, also on this one, yeah, here I do have to play a bit more with it. This one's. Yeah, now it's better. Also for the channels. And we have also the 3D plot in here. So if I go directly to this line, we can see the signals for all of them, for every single case. Source detector, source detector, source detector. Okay. And here are also the block averages, where you can see that all the signals are quite nicely differentiated. Now I will switch back to the line plot, stop it for the moment, Stop also the paradigm and go to MATLAB Simulink. Sorry, I had to close the paradigm presenter because it was just in front of my screen. I will reload the paradigm, which is on desktop. Yeah, here's paradigm. Paradigm is not imagery. So start. So now I will open the Simulink model. This one here. I will shrink a bit our paradigm presenter so I can nicely see. Okay, so I will start the paradigm again and we'll search for the amplifier. G.Nautilus, select. I will apply exactly the same filters also in here. So band pass 0.1 to 60. And 48 to 52. Okay. Now, we do have to check the fNIRS. So I will start now the fNIRS data acquisition. Okay. And now going back to the model. So we do have in Simulink, we do have to check the stream availability in HISIS. So click on it. And we get this message stating that the stream can be solved on the network. Okay. And then the marker ID. Again, also in here, we have the marker stream name, the marker values, and let's check also for the stream availability, which is in here, and it's present. It's okay. Okay, again. And now I will start the recording. So as I said, HISIS records both fNIRS and also EEG. I will show them to you in the plot in a bit. Okay, it started recording. So this is the HBO scope. And this is the EEG scope. I will put them both on the screen. And here, as you see, we have the cue for pointing to the left, telling to the user to start imagining the left hand movement. And we can see both markers also in both EEG and in HBO. And in fNIRS. Okay, auto scaling again. So in this case with HISIS, HISIS saves this Simulink model, saves all this data, both EEG and fNIRS data, under a MAT file that can be then processed from MATLAB and doing all the processing that you do want to do. Okay. So Alex, you could start, I don't know, imagining the movement, just following the paradigm. We don't have time now to perform a full experiment, but yeah, it is, maybe we can see something in here. It's not the best plot. Let's add also Aurora. In Aurora, you can see it nicer. All of them work at the same time. Just relax. In a couple of moments, we'll see. Look at the cube again, and please start imagining right-hand movement, even to execute it. It will help a bit. Yes, we can see in here a nice increase in this channel. Okay, so as I told you, these are the ways to record both EEG and fNIRS. And going on now, I will show you these results, some other results from a motor imagery experiment that I did previously. So in here we can see the averaged HBO and HBR during for the left-hand trials. In my case, so we can see that they are very nicely distributed. So the peak in here and so on. And for EEG, the results, so these are the common special patterns. So common special patterns method is very well known for discriminating in between two populations, in between two classes. So in here we have exactly the electrode disposal, like Alex says, and this blue spot in here represents the location where I had the event-related desynchronization while I was imagining the right-hand movement. And in here in the lower right side, we have the spot while I was imagining the left-hand movement. All these plots can be nicely seen also on a 3D brain, on a 3D plot in here, so on the scalp directly. This can be moved to the right, to the left, and so on. And also directly projected to the brain locations. So we see the electrode as these black spheres that are up and beneath the projection on the brain. Okay. Now, the last part of the presentation. So first I will stop this here for the moment and also the recording. And I will show you how to dismount everything.
So I think you have a raised hand.
Where's Zoom? Here, I cannot for the moment because I'm sharing the screen. Okay, not from the team. But yeah, after, let's say that we perform the full experiment, now I do have to put everything down. So it's very easy. First, as I showed you earlier, just remove the clips nicely so they won't get gel on them. Put them all on the table. As soon as I'm removing a optode, I'm putting it in the back where there's no EEG, where there's no EEG gel. Okay. This is the undoing procedure. And after this, the cap is ready for being washed. So the left side is done. And now I'm doing the right side. So now all the optodes are off. Nicely here, gently on the table. And now I can close all the measurement. I think I stopped already. The Simulink, stop the amplifier, and start preparing for the nice words from my colleague after removing the cap with the very nice hairstyle that he has now. So Alex, thank you very much.
You're welcome.
And let's switch places again so I can start answering the questions.
Thank you very much, Danut. One of the questions is which types of classifiers are you using for EEG signals?
Right now we are using LDA classification.
Any reason for that?
No, because it's very fast and pretty accurate.
Then Johannes is asking, is a combination of EEG and fNIRS also possible in a portable setup with less setup required?
It is possible in a portable setup. So this the fNIRS device, it is quite big, but it's battery powered and it has the opportunity to be connected to the PC via Wi-Fi or Bluetooth. So yeah, only that the user has to carry it in a small backpack, and yeah, the rest it's possible in a portable way.
Maybe on the Nautilus webpage, you can also find our fNIRS sensor device, which is an 8-channel fNIRS device, and you can just put it onto the Nautilus more or less, and then everything is sitting on the head. That's even smaller, but it has only 8 channels. Then somebody is asking, is a hybrid fNIRS and EEG recording the best way to get meaningful BCI recordings?
From my point of view, yes.
Any reason for that? What do you gain?
As I said earlier, so it's a complementary part. There are many people, for instance, in motor imagery, who do not have very good results in motor imagery from the very first time, so after the second or the third session. But in motor image, together with fNIRS, we could have it. So checking the oxy and the oxyhemoglobin, those changes can be very meaningful for the classifier.
Cheng wants to know, is there any interference between the fNIRS and the EEG? What are the problems that you get when you're doing both?
No, I never had interference in this case. So there aren't, because there are two different principles. So fNIRS measures the oxygenated, it uses the HBO and HBR. So the EEG electrodes measure neural activity. So they are measuring a very small voltage. So there are two different things and do not interfere with each other, not at all.
So there's also no electromagnetic interference. This could occur, of course, if you're using two different electronic devices, but this is not the case, so they work nicely together. What's about the spacing of fNIRS and EEG? So you have to separate them, otherwise you get shortcuts, the liquid goes into the optodes and so on. So each considerations are important.
So we do need something like 1.5 centimeters between, or 1 centimeter between the EEG electrode and the fNIRS dot.
So important is that the EEG electrode has a little reservoir, a little ring underneath the cap, and when you squeeze in the electrode gel, then the reservoir keeps the liquid inside. Otherwise, it would float around and it could float to the optode, and then you would get the liquid into the optode, which can be a problem. And the electrode gel also makes the contact between the EEG electrode, made, for example, of silver chloride material, and the skin. In the case of the optode, the light has to shine into the scalp and into the brain. And it's important that you remove the hair beforehand. And this means if you remove the hair, you must be careful that you're not putting it under the EEG electrode and vice versa. So there's a separation required, And this also means that you cannot precisely record from the same position. And important to know is also that the EEG electrode records the voltage just below the electrode, of course, its voltage. And the fNIRS records from the sensor or from the sender to the receiver. So there is a certain distance between them, and the distance guides actually how deep the fNIRS signals goes into the brain and how deep you can do the recording. So if you want to record more or less from the same brain region, then you have to put the EEG electrode exactly between the fNIRS sensors to be located there. Subashini is asking, could you please mention new applications for EEG trainings for EEG to train new skills?
EEG to train new skills.
So I can also jump in here. So we showed, for example, today Recoverix. That's a kind of neurofeedback closed loop application where we can train fine and gross motoric skills with EEG feedback. People have to imagine left hand movement, right hand movement, foot movement. Then we stimulate the hand and people get better control. This helps also for spasticity. So we see reduction of spasticity when people are doing that. And they can also swallow better. They can speak better. So there are a lot of interesting side effects. One of these side effects is also better concentration performance and better memory, which is of course also very interesting. And then Mahima is asking, in the previous presentation, Frankie used Oscar, and the eye blinks were completely removed from the normal EEG. Why is this? Danut, you want to answer?
Yes, because we do want to have the clean signal. We see the eye blinks as artifacts, and that's why Oscar removes them. Basically, we do not need the eye blinks.
Yeah, that's an artifact for us, so we don't want to have them. So for BCI applications, eye blink are not really a problem because they have a different frequency range, so they are outside of alpha, beta, and of course gamma, and they occur very seldomly. But still, it's easy to remove them because they are so distinct. Then Mozart is asking, what are the necessary considerations required for light intensity for fNIRS beside hair density and skin thickness?
So these are the only ones. Basically, with fNIRS, you can set up, if you still have a bad signal, you can increase the source power of the LEDs. So that's it for this, in this case.
So Oscar is of course removing artifacts and making the EEG more stable, but which other signal processing do you need to run a BCI experiment?
The signal processing for a BCI experiment. So basically it's filtering, extracting different features, like for instance in motor imagery, we do first filtering it between 8 to 30 hertz, then spatial filtering this data with the CSP filters, then doing the normalization or log transform, then all these features can be done, can be fed to a classifier. So normal filtering, then spatial filtering, normalization, log transform, and then LDA applied.
Maybe Danut, you can also put your email into the chat. So if people have more questions about EEG, BCI experiments or fNIRS, so they can reach out to you.
Sure.
And thank you very much for the nice presentation and the nice experiment. Also, thank you to Alex for being the subject. Very well done. And enjoy the rest of the day. Thank you very much.